Morning, you guys.
Good morning.
I'm just going to manage the waiting room for a few minutes. If you can hear me, we're going to start right at 9:30, so another minute or so. All right. I'm just letting the last few people into the waiting room. I'm sure some other people will join us later. So good morning, you guys. You're all muted right now, so if you have specific questions, just remember you're going to need to unmute yourself. For right now, I'm just going to start us off with some of the first stuff we did last week, and we're going to review a little bit of that and then work our way through the activities. There were 3 activities we had you guys working on last week. The first one was a continuation of the week before spring break. Good morning. And what that was the continuation of the genetic stuff. So we'll start with the more complex genetics, since at this point I've already spent a little time with the PBS group reviewing basic genetics. And then we're going to transition to just quickly looking at the connection between malaria and sickle cell disease and make sure you kind of get the key takeaways, which it looked like most people did, but some people had missing segments of their notes where it really would have communicated to me that you truly understood. So I want to just kind of review that with everybody. And then we'll talk just briefly about the difference between sickle cell disease and sickle cell traits. And then we can revisit some of the characteristics that you shared out in your Flipgrid video for the NCAA athletes. And then if time allows, I'm anticipating going through those second and third things pretty quickly. So if time allows, we can talk about heart stuff, since it's not last week's work, but we have the time, so we might as well look at some of the things you guys have been looking at this week. So I'm going to go ahead and share my screen, and for whatever reason right now I'm staring at Haley Keller. Hi. So I'm gonna go ahead and transition away and bounce to a component of the 3-4-2 activity. So what I thought we would do is take a look at how you'd come up with an answer to the pedigree that you see on the screen here from 3-4-2. A few of you have watched a video that I prepared where I talked through how I would figure out the genotype for Anna's mom using the pedigree. And I would consider this pretty basic genetics. This is not a complicated type of problem that we're going to be working through here. So first of all, I'm going to go ahead and open up my iPad to kind of walk through the problem. And I'm going to try and see if I can't get both my iPad screen and this pedigree up kind of simultaneously. We'll see how this goes. So first things first, let's get it so that you can actually see my iPad. Hold on one second. You cannot see what I'm typing. That's annoying. Why not? My kids use my iPads when I'm not paying attention, and sometimes they disable the ability of like the pen to work and stuff like that. So I might not have the ability for it to work, which sucks. Tech issues. Oh, I bet they have been using it and then didn't charge it. Shoot. All right. Well, with that said, what I might have to do is make like a improvised document camera and join my own Zoom. So give me 1 second. I'll use my phone and I'm going to join this Zoom conference if I can. I haven't done it this way yet. Do you guys know if you have any teachers that are trying to join their own Zooms and use their phones as a document camera? I've heard it works, so we'll see. We might get some weird like background noise. So then let me think. All right, hold on. Gotta think of how to do this. Okay, now you're staring at my phone. All right, let's try this. I'm gonna open up my camera and I'm gonna use it with a piece of paper underneath. Can you guys see my, you see my phone, yes? Okay, see how this works. Meanwhile, I'm probably gonna get like messages bouncing through from other people that are messaging me on my phone. So as far as the pedigree goes, I would start by considering who Anna's family members are. And so let me get this aligned. Be nice if I can make it bigger. I don't know that I can, so we're gonna have to go kind of small here. Get that off the screen. So we're gonna go ahead and draw out Anna's parents. Whoops, that's sideways. Anna's parents. Okay, and then we know that this is her mom. I think her name's Maria. We were told from earlier activity that her dad was a carrier for sickle cell disease. So we know then that his genotype is that one. And then Anna obviously has the disease and she has a brother who does not and doesn't even have a carrier status that was derived from gel electrophoresis. So this brother has that genotype right there, 2 capital S's, 2 of the dominant alleles, and in the pedigree he wasn't shaded. I believe that's Eric. Anna herself has the disease, so we'd completely shade her in, and then her genotype would be 2 lowercase s's. And then she has this other sister, Juanita, who is married to Jason, and together they have a daughter, Erin. So Juanita is without the condition. Whoops. Juanita is without the condition, but I believe the gel electrophoresis told us she was heterozygous. Let me verify that that's true. I should have the pedigree. Yes, that's correct. Okay, so Jason, we figured out earlier, is, I think he's a carrier too, and then his daughter, Erin, has the disease. No, Jason has the disease too. So he's completely shaded in. All right, so we got lowercase s's for Jason. So now all we're trying to do is backtrack and figure out what Maria would have been. And a lot of you did it, but perhaps you weren't really aware how you did it. I had posted an answer key with the answer. We're gonna find that she's heterozygous as well. And the reason for that is as follows. For these two parents to have given birth to a child like Eric, each of them had to be able to pass on a dominant allele. So each of them has to have a dominant. And then in addition to that, in order for them to have a child like Anna with two of the recessive alleles, each of these two parents would have had to have contributed one. So therefore, each of them would have to have that, giving us the knowledge that Maria is a carrier. If you're asked to show work to kind of explain yourself, then you could do one of these Punnett squares where you put Maria's letters. You know she doesn't have the, well, actually, I don't think we even knew she didn't have the disease. We didn't really know anything about her. But we did know the dad didn't have the disease. So we would have this kind of arrangement on the side for the dad. And then in the mom, we could have originally had blank blank. And then you know that you can fill in whatever's possible and keep these little blank lines in place. And that kind of helps you fill in the missing details. So we got to have Anna. Got to have Anna in this Punnett square, and we also have to have her brother Eric represented. So again, the thinking being, the only way you get an Eric is if one of the parents had a dominant allele and the other one did. And then the only way you would get an Anna genotype is if the mom also contributed a recessive allele. So you're sort of working your way backwards through this problem and figuring the scenario out, okay? So that was kind of the first big thing I wanted to look at today related to complex genetics. And then the second one, I'm not gonna go over unless anyone specifically wants me to, but basically no, I see double. What we are looking at is the second conclusion question to activity 342. And I had a student last night who had, for the 20th time, questioned what the answer was. And so I actually made a video that kind of went over why the answer is what the answer is. So I had previously included the answer key, but then I included a video link. And I just sent it to that student alone. But if you want it, let me know and I can share with you, I wrote request. I can share with you the video link to 342 number 2 conclusion question so you can see why the answer is what the answer is. It was that kind of convoluted one where we had to like look at Gina and Juan and what the, or whatever, Gina and Juan I think are their names, and what are the chances of them having a child with sickle cell disease. So from there, unless someone has a specific question about those specific things we just went through, I want to take a look at a couple other types of complex genetics, if that's okay. Are there any objections if we do that? And I'm charging my pen, so maybe I can use my Apple Pen in just a little bit. It does say it's got 1% battery life, so that's why it wasn't working. I'm gonna go ahead and try and take my phone and like make a document camera by propping it up on something. So give me 1 second. I'm gonna try and pull my kids' names here and see if I can give me some height. This is what I used the last time I did this. This is called at-home improvisation. All right, let's see. sideways. I'm finagling boxes here. I know you can't see that, but that's what I'm doing. That might work. I just have to figure out a way to hold it upright. I don't know how this is going to work. You guys should see my setup right now. I have a game, a coffee container. It's being annoying about my phone. It won't like let me turn my phone, otherwise it turns sideways for you guys. Teaching from home is fun, let me tell you. I'm being sarcastic, by the way. Okay, let's try this. All right, so I thought we could try a problem that's like a sex-linked problem, and they're a little bit more complicated. And in addition to that, we can also tie in like one of the other non-Mendelian concepts. So I'm just going to pull one randomly from our bio book since I have it nearby. I know not all of you are in biology, so you don't have access to this if you're not. All right, I'm going to kind of make up a scenario to go with one of the ones that's in my workbook as well. So let's go ahead and I'm going to combine a bunch of things at once. I'm going to combine, if I can do it, I'm going to combine sex-linked as well as a dihybrid problem and codominance. So it's going to be like 3 different patterns of inheritance all within one problem, okay? So if this stays focused, let's come up with a scenario. Let's say that we're going to explore chickens. Okay, and chickens can have different kinds of feathers. Let's say they can have black feathers. They can have white feathers, and I'm going to switch letters to communicate the difference here. And then they can have feathers that are both black and white. So making a key for this characteristic, I'll use B's for black. I'm going to switch to the letter W for white, and then combine the 2 letters to show a chicken that has both black and white feathers. So this scenario, because we're seeing both colors here in the heterozygous type state, it's not technically heterozygous. You're actually seeing both dominant at the same time, and that's a pattern that we would refer to as codominance. So we have codominance happening here, which is why I use 2 different letters of the alphabet, and I'm capitalizing them both, trying to communicate that they're both equally dominant at the same time. So we're going to combine that characteristic with a second characteristic of chickens, and it just so happens that that second trait we're going to combine is a sex-linked. So let's say on chickens, let's just stick with like hemophilia. They probably have something similar to that in their own blood. So let's say that hemophilia in chickens is carried on the X chromosome, and it is a recessive condition. So female chickens can have this genotype, which would mean that they do not have hemophilia. They clot their blood normally. If you remember, hemophilia is a condition where you do not clot your blood properly. My camera times out after a little bit. And then we have a female here who could be heterozygous for hemophilia. So we would say she also has normal blood clotting. And then you can have females that are homozygous recessive, which gives them hemophilia. In the case of the male, they only have 1 X chromosome. So they would have, excuse me, 1 X and 1 Y. And in some cases, they can inherit the dominant allele, which gives them normal blood clotting, or they can inherit the recessive allele, which gives them hemophilia. Okay, I'll just write hemo. All right, so let's say we have a male chicken and a female chicken, and they're going to mate and have babies. And the first chicken is a black and white feathered chicken who is female, and she does not have hemophilia. Now, she could be heterozygous or homozygous dominant. I'm not sure. So on her second X chromosome, I'm just going to put a little placeholder, which is this little line. Her spouse chicken is going to be white feathered. Yeah, chickens get married, don't you know? And then he's a guy, so we'll do XY. And let's say that he does have hemophilia. So we'll put the little H on his X chromosome. So now it looks kind of crazy, and the steps of doing a dihybrid problem are going to apply. So we need a FOIL. FOIL is, again, where you take the first letter of each of the two traits and combine it. For the F, so we're going to take our B and combine it with our X that has the capital H on it. Then I'm going to take the outside letters, which is this B and this X, and combine them. Even though we're unsure of the letter on the X, we're going to just leave it like that. The next one is the inside letters, so the W, and then this inside X. And then the last letters would be the last W and the last X. Looks kind of crazy. As far as her husband chicken goes, it's either going to be a combination of W with the X and the H, or a W, and keep in mind these W's are capitals. We don't have lowercase W's in this scenario. And then it would be a Y, and he'll make 2 of each kind. If I could FOIL it if I want to, but that's going to be the end result. So then we set up a 16-box Punnett square, super fun. And I'll try and squeeze all 16 boxes on the screen here and legible. We'll see how that goes. Okay, and so I'm going to go ahead and put the female's genes across the top. That's one. That's the second one. That's the third one. And that's our fourth one. Over here, we're going to do X, the little H, W with a Y, and then it repeats. Again, down low. Oh, you can't see. Sorry. Can you see? Or did something come across? Looks like my iPad is trying to mirror as well. Okay, so filling this sucker in, keep your B's and W's together. It's going to make it a lot easier later on. So BW all the way across, BW, WW, and WW, and then now we deal with the X's. So this one's going to have 2 X's with capital H's attached to both. The next one is going to have 1 with a capital H and 1 with a blank. The next one would be, I'm running out of room, 2 capital X's with H's on them. And this last one, give myself a little bit more room on this side here. Messy. This last one is the X with the capital H from over here, and then the X with the placeholder. All right, so it looks a little crazy. Let's do the next row, and then we're basically done because it would have repeated itself. So BW, X with the capital H and a Y, so we're making a male chicken here. BW, X underscore Y. WW, X capital H Y, and then last one WW X underscore Y. My phone is set up to automatically try and scan things, so sorry that little button keeps popping up that says scan. Okay, so again, that repeats itself. So basically at this stage, we have to interpret what we have here, and I'm not going to do genotypes because that's chaos. Let's just look at what they would look like. So this particular one would be one of those black and white chickens, and it would not have hemophilia. It would also be female. This particular one would be similar, not have hemophilia. Could be a carrier, but we don't care. It doesn't have hemophilia. This one is a white chicken, that's 2 W's, and a female who does not have hemophilia. And this one is a white chicken, female, who does not have hemophilia. These are all the females that these parents can produce. So they will never produce a female who has hemophilia. They will have an assortment of colors, black and white combo and white feathered. They'll never have a black chicken. Then we come down here and it's something similar. You have the black and white feather male who does not have hemophilia. Here you have a black and white male who may have hemophilia. It all depends on what mom was contributing on that X chromosome that's unknown. This is a white male who has no hemophilia, and this is a white, sorry, I said female, I meant male. And this is a white male and may have hemophilia, we don't know. So there's still some unknowns, but if the question said something like, what are the odds of these chickens producing a female with hemophilia, your answer would be 0. They cannot do that. And the chances of a male, well, it could be that of their males, half of them have hemophilia and half of them won't, but it might be actually a 0% chance depending on what mom's contributing. So if somebody asked you as a genetic counselor, because you know chickens, they go to genetic counselors all the time. If somebody said, what are the odds of these chickens having a male with hemophilia, you would have some uncertainty. So you'd have to say, well, it's up to a 50% chance. But it could be as low as 0. And in order to get clarity, could be 0. In order to get clarity, that female chicken, we would have to know a little bit more about her. So maybe we find out, like, is there any hemophilia in her past? If we know what her family, her parents specifically were contributing, maybe we figure out her second allele. If that still isn't enough information, then a person could go forth and actually get like DNA sequencing done. So maybe a chicken would choose to get DNA sequencing done. The point is that you may still be in a situation where you don't know. And that's pretty much how it ends up with the conclusion question for the 3-4-2 activity, where you're a little uncertain, so you have to give odds. And some of my students I saw are going real crazy, and you guys are doing all kinds of probable math calculations. You're like, well, it's between 0 and 50, and so it's actually 32.5. You can just give me the range, and that's fine. It shows me that you're thinking through the process, and like you're coming up with, well, it could be this or it could be that. It's somewhere in between, but without more information, we don't totally know. Okay, so I'm going to pause for a second. If you guys have some specific questions about this crazy problem, that's probably the most complex problem I can think of, combining like 3 different things all together there. Okay, nobody's interjecting. Maybe you're not bold enough to do so this morning. Okay, so if that's kind of it for the complex genetics stuff, which is obviously already graded, we're going to go ahead and move into the malaria piece. So I'm going to go ahead and mirror a different screen, which I think means I need to change something here real quick. Give me 1 second. I think I can no longer be our Zoom host. Okay, that's weird. You guys saw all of that, yes? Okay. Can you see me now? Can you see my computer screen with a pedigree on it right now? Okay. I think I just disconnected my phone, not realizing it, which is fine. So let's go ahead and bounce to the 3-4-3 activity, and I'm just going to do a quick review. We're not going to do a lot of time on this. This is the one that you did last week related to malaria. And so I do realize that very often the video clip from PBS that was linked inside the activity right here does not open very well. Oh no, sorry, the second one, a mutation story, I think it is, does not work very well. And so I had included a separate video that I think a lot of you did visit. That's kind of where most of the content comes from. And that is coming from something called HHMI. Okay, So basically what they explore is how, when you look at the map of hemoglobin disorders, you should have noticed that there's a heck of a lot of hemoglobin disorders in Africa. I think my map is opening right now. Specifically in like the middle band of Africa, it's the darkest, which means it's the highest quantity of different types of hemoglobin issues. It's not just sickle cell, it's other hemoglobin issues as well. And then over here in South America / Central America, we have a higher incidence as well. And so that should tell you something. We got to think about what's common to those places. Those are warm locations. They're basically right along the equator. And so they also are tropical in those locations. And so what will happen is that because it's tropical, you guys are able to see the map, yes?
Not anymore.
Not anymore, but you did a second ago?
Yeah.
Okay, Zoom sometimes confuses me. Anyway, because it's tropical, that means that different types of organisms can breed there, they can thrive there. And one of those organisms is a particular mosquito called the Anopheles mosquito. The Anopheles mosquito carries a protozoan in its saliva, and that particular protozoan is the one that causes malaria. So what will happen is basically the malarial parasite or protozoan, whatever you want to call it, is injected into the skin and the bloodstream of a person who is bit by the mosquito. It will continue its life cycle inside the red blood cells specifically. And that's where it goes, and it will replicate and pop out more malarial parasites within the host's body that can then breed eggs, become a little mosquito carrying thing, and it will go off and infect other people. So what will happen if someone has sickle cell disease is that because their hemoglobin is misshapen in certain environmental conditions, if it becomes sickled, which will happen when the parasite is injected, it will sickle, it will trap the parasite inside, and then that parasite cannot reproduce. So that's actually a really great thing for that person because they're never going to really get sick from malaria the way that someone otherwise would. But obviously they have sickle cell disease. And if you think about that map and the regions of the continent or the regions of the globe where we have higher incidences, these are poorer areas and they don't tend to have as good of healthcare. So in these regions, a lot of people will succumb to their sickle cell disease. So they won't necessarily be alive for very long with sickle cell disease, even though the disease itself protects them from the malaria that's also deadly. But on the other hand, somebody who does not have sickle cell disease, who has a homozygous dominant genotype for sickle cell, will never have cells sickle no matter what, and they are totally vulnerable to the malaria. And so living in that environment, if they don't take precautions, they don't keep the nets, they don't spray, yada, 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 they're probably gonna succumb to malaria at some point in their life. So who's got the evolutionary advantage in this scenario is the individuals that can sickle their blood and therefore can trap malarial parasites and not get sick from it. And those people are people who have sickle cell trait. Sickle cell trait is just a way of saying that someone is a carrier for sickle cell disease. They're heterozygous. They don't generally have symptoms of sickle cell disease. And that's because about half of their red blood cells can potentially sickle. The other half are totally normal hemoglobin. They won't do the whole polymerization business that we explored a few minutes ago. So that said, some people have symptoms of being heterozygous, but it's only going to be brought on in extreme conditions. Extreme conditions would be something like sports. And that's why we had you guys do a little work with the NCAA athlete recommendations, okay? So without getting into all of that just now, people who have sickle cell trait have an evolutionary advantage living in these environments. So you've got people who are the extremes that are going to die from their conditions. One extreme, I have sickle cell disease and I'm going to die from my sickle cell disease because the healthcare is not good in my country. The other extreme, I can get malaria and it's going to kill me. Then you've got that middle range where people have the trait for sickle cell. They themselves don't have the sickle cell disease, but they do have protection from malaria in about 50% of their cells that can potentially sickle. So the malaria doesn't really cause as many problems for somebody in that state as someone who didn't have protections at all. All right, so over time, That's been the pressure from the environment in these regions. And thinking from an evolutionary perspective, putting on like your evolutionist's hat real quick, it's an advantage to have that condition, that genetic status for survival in that environment. So over time, the environment has encouraged individuals to be born with that gene because they're the ones that stick around, they're the ones that reproduce, they're the ones that pass on those genes to their offspring, and we continue to see the sickle cell gene popping up in those populations. Now, you guys know basic Punnett squares, so you know that somebody who is a carrier for sickle cell disease can easily have children who have sickle cell disease. You've got people, let's see if the pen is charged enough to do a drawing. You've got people who can combine two carriers, two survivors in this environment, can have children and have a 25% chance of having children with sickle cell disease with every pregnancy. So no matter what, these parents are survivors and they can stick around in Africa and some of these other populations that have malaria, but then they pass on the disease 25% of the time. So we continue to still have people pop up in these populations with a deadly disease that will kill them in those geographic regions. It's a lethal gene in those regions. In the US, it's not really lethal because we have good medical care for sickle cell disease. But that said, this is why this persistent lethal allele keeps popping up, because we have a heterozygote that has an environmental advantage, an environmental adaptation, we could say, that proves to be beneficial for survival. And that is all about evolution. That's why it's evolved to be so common in those regions of the world. Okay? If you don't have the malarial parasite in your environment, then it's like it's non-existent. That particular environmental pressure is not a thing. So when we look at the map from the United States, like our incidences are not so high, they're pretty low. And the only reason it's gray as opposed to being just pure white is because we have immigration here. So we get individuals from these places coming here. If you look at Canada, it's white. If you look at Alaska, there's not a lot of people immigrating to Alaska, so you don't have a high incidence there. Norway, Sweden, Finland, over here in Northern Europe, they don't have a lot of immigration, so you're not going to see as much of that. Not to mention those are colder areas too, so definitely the mosquito does not thrive there. But the mosquito is not found here either. We're just higher than we otherwise would be because of immigration. Okay? Anyone have any questions about the takeaway from that activity? Some of you were cute and you went and did the stuff with the plastic cups and the beans. You didn't actually need to. The instructions were just to take notes. That's it. I don't expect you to go do labs like this on your own time. You're not going to go do a sheep dissection at home, right? Okay, so then the last thing last week was just a quick connection between NCAA and the athletes who have sickle cell traits. A few students, take a look at the Synergy gradebook if you're unsure. A few students made their Flipgrid video where they talked all about sickle cell disease and how it can be scary for people who are athletes. That's not what the topic is about. The topic is about people who don't have the disease, they have sickle cell trait. So that means, again, that they're heterozygous carriers for sickle cell. About 50% of their red blood cells can sickle. They only sickle in certain environmental conditions, and one of those is hot weather, one of those is elevation, one of those is oxygen deprivation, such as during heavy exercise. So if you're a college athlete, you might very well be doing a few of those conditions in your time. And so the NCAA, the colleges, are very aware of those who have sickle cell trait could be symptomatic and have problems with their athleticism. And so that's where you guys went with the Flipgrids. The recommendations are really no different for a sickle cell trait athlete versus someone who actually was athletic and had sickle cell disease. My guess is there are not too many people that are athletic at the college level who have sickle cell disease because the disease is too debilitating. They wouldn't be able to, do their activity. But for the most part, the recommendations are the same. Even if your video kind of went sickle cell disease direction, your recommendations to your patient weren't really any different. All right. And then again, last week, you guys went and did Unit 3 assessment on Socrative. Just FYI, I can see how many times you take the test. So some people took it once and they got like a 10%. I'm like, okay. And then I see you take it a second time and you get a 40, and then you take it a third time and you get a 70. I'm like, So I can see all of that. So the next time you do those, you know, be aware. I can see that. I can see that on the quizzes as well if you take it multiple times. I'm not sleuthing you. I'm just letting you know. Okay, so that's it for last week's stuff. Are there any questions about any of that content that anyone wants to ask for the greater good of everyone that's together right now? Okay, I'm gonna go ahead and stop sharing my screen for a moment. There we go. And then I'm gonna go ahead and open up a diagram of the heart, and we can take our last 20 some minutes to take a look at some of the heart stuff. I'm curious if you guys want to unmute yourself. What are you thinking about the way I had to do the assignment this week? Do you like the format of having, you know, like a separate document with all of the steps to accomplish? How did the animation thing go? I've been grading them and they look pretty good.
I thought the animation was like interesting. Like I didn't hate it, but I didn't love it.
Just trying to like drag arrows around and stuff to make it look correct.
Yeah, and my computer sometimes gets like really touch sensitive, so like it'll be like
oh, so that was annoying for your device. Well, the reason I did that rather than just having you write out the blood flow is I thought, you know, you could easily copy and paste the blood flow from some other resource, but actually making like the slides yourself was an act of, do you know what you're doing? So I was hopeful that would give me a little bit more information about what you guys are understanding. So it looked like pretty much everybody was doing really well with the heart anatomy that has submitted their work already. And I really wish I could draw on my screen. Let's see if this pen keeps up today. I might be able to do exactly that, and then I'll share my iPad screen with you guys. All right, so why don't we review the heart anatomy while we go through the flow of blood, and then the heart dissection is just not an option to replicate without being with a sheep heart. And so unfortunately those YouTube videos and things that I've linked you to are basically like all you're gonna find. See if I can do unlabeled. There was a teacher who teaches this class who went to his classroom and got his sheep hearts and brought them into his kitchen and then did like a live dissection with his students. And I was like, well, that's kind of fun, but at the same time, like, I don't know that you're gonna get anything more from me doing that than if I was just giving you a YouTube video to watch. Because that's the same thing, right? So anyway, I'm not bringing sheep hearts home. My kids will have a cow. All right, can you guys see my heart diagram? I think you can. You can do head nods. Okay. All right, so I'm gonna go ahead and kind of walk through the blood flow pattern using this picture. And then I'm gonna write on my screen if my pen cooperates and just sort of label the heart parts at the same time. All right, let's see how this goes. So we're gonna start at the top where the blood first enters the heart, and that's gonna be either right here or at the very bottom, which is actually all the same tube. That's one long tube right there. And you see how it's colored blue? That's misleading. I understand that sometimes middle school teachers will educate their students to think that blood that isn't full of oxygen should be blue. It's not. Your blood is never blue. Your blood is always red. When it's fully oxygenated, it is very bright red. You know that because you bleed from a wound and you have to put Band-Aid on it and it's bright red. When it's less oxygenated, it's like a deep purple. So it's not gonna look blue ever. In your veins, it might look blue because of the coloration of our melanin in our skin. So there's not actually blue blood in there, okay? So they're going to communicate to us what's deoxygenated with blue in these images. Deoxygenated implies that the blood has no oxygen. Deoxygenated sounds like there's no oxygen. In reality, it's never without oxygen. It just has less. So this is going to be blood that's coming from the body where it's already had all of the nutrition and oxygen removed. And now it's going back to the heart so it can, 1, pick up oxygen at the lungs, and 2, so that it can then be pumped right back out to the body so it can use the blood and its nutrition and oxygen again. So we're going to start with the top end of this tube. This is a vein, but it's known as the vena cava. And the top end, we use the term superior to refer to it. Okay, superior implies top, like you're above all else. Vena cava down here is referred to as the inferior vena cava, inferior being down low, okay? So typically superior vena cava brings blood from like the top half of your body. So if you think about where your heart is located in your chest, everything from that, like your shoulders, your arms, and your head, all of that blood is going to come in through the superior end of the vena cava, and all the blood from the bottom half of your body is going to come in through the inferior. In both cases, the blood enters and spills into this chamber right here. Now, keep in mind, when you're looking at a heart, you're looking at it as it's in the person's chest, so it's actually mirrored. So what that means is that the side I'm on right now is actually the right side of the individual. And then over on the other side is our left. So we're going to enter the right side of the heart first. The top chamber is actually not as large as it looks. It's actually pretty small relative, and that's known as your atria. So I'm going to label the right atria. Sorry, it's atrium. Atria is plural, is right there. And then the blood will move from that atrium and it goes down with gravity through this valve right here. The valve that's on the right side is known as the tricuspid valve. And you'll see it has 3, well, it kind of looks like more than 3 cusps, but there's basically 3 cusps, 1, 2, 3. And there's a little like muscular projection. This picture makes it look like there's 4. There's a little muscular projection, and what's happened is, I'm going to get rid of these little diagrams I just drew. There's this little fibers that are in here that actually pull on a flap that's in the center of the atria and the ventricle, which is just beneath it. And that particular flap is capable of opening and closing, and it only opens and closes one direction. So imagine it being like a flap of tissue like this, and then those little muscles and fibers pull it down to open it up, and then they snap back closed. So these little projections down here are muscle. They are a special kind of papillary muscle. Papillary is all the muscle in the heart. All cardiac muscle is papillary muscle. This right here is also a type of muscle in the heart called myocardium. And if you look around the heart, you see it everywhere. All of this is myocardium. Okay, it's kind of like the beefy part of the heart. That muscle is what gets the heart contracting and expanding, pushing the blood around. Let me backtrack and get rid of all my highlights. These little papillary muscles down low, oops. These little papillary muscles down low are pulling on these little tendrils. These tendrils have a name. They're known as the chordae tendineae. We're speaking Latin now. Chordae tendineae. And that's these little tiny fibers that you see here that pull on papillary muscles to open and close the valve, okay? So this particular valve, again, is known as the tricuspid valve. And I always teach my students to just think of the letters T, R, and I rearranged to spell right. R-I-T kind of sounds like the word right. You're on the right side when you're dealing with your tricuspid valve. Haley, are you dancing? After the blood is in the right atrium and passes through that tricuspid valve, it spills into the right ventricle. The right ventricle is all of this space. So that's a pretty sizable chamber, okay? And at that point, all that blood is pooling there, waiting to get pushed out. And it's gonna get pushed out through another valve. That other valve is a little semi-lunar valve. It kind of looks like two crescent moons. And that's known as the pulmonary semilunar valve. Anytime you see the word pulmonary, it references the lungs. So we have lungs on both sides of our heart. We have a left and a right lobe of our heart, of our lung rather. I'm going to draw those in lightly here, okay? This is the left lobe of the lung, and this is the right lobe of the lung. And our body is efficient, and we're going to pump blood to both simultaneously as long as both lungs are functional. So right now, the blood has moved down and it gets pushed up from the ventricle through this little valve right here. It's known as the pulmonary semilunar valve. And semilunar refers to its crescent moon shape. There's like two little crescent moons. There's actually three, and they kind of come together and form this little closed door. And the door can open with enough pressure, and it will flow only upward. So the door goes snap upward, lets the blood flow up, and then as soon as the blood flows up and it's out of the ventricle, those doors should slap shut, okay? And the same thing is true of the tricuspid valve. It should shut closed after the blood has moved. Now I asked you guys to explore what happens if a valve is not functional. That's called a prolapse. And so what can happen is the valve can open, shut, but not shut all the way. So you basically get blood that kind of like squeaks its way through when the doorway should be shut. And that's usually audible when a doctor is listening to heart sounds. They can listen to the heart go lub-dub, that's kind of the beat, lub-dub, 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 and they'll hear like a whoosh sound as the heart fills with blood and then pushes its blood. And what they'll hear is a lub-dub, squeak, lub-dub, squeak. And that little bit of squeak is just a little bit of blood like pulsing itself right back up into the spaces where it isn't supposed to be anymore. So that said, there's a bunch of repercussions that can happen. Usually it's pretty minor. We call it a heart murmur, by the way. So if they say, oh, you have a heart murmur, it usually just means there's a squeak. And so one of the valves is weak. It could be weakened by a virus. There's childhood viruses that a lot of people get, scarlet fever being one of them, that can weaken the mitral valve specifically. I don't know why not the tricuspid, but the mitral valve is more prone to that kind of weakening, I suppose. In some cases, it may be genetic, okay? There's a variety of things that can cause it, but that particular valve, because it does what it does, it actually makes someone slightly more prone to an infection of their heart tissue. So for example, I have a very mild mitral valve prolapse. Sometimes the doctors say, no, you don't, because it's that mild, they don't even necessarily detect it. But apparently when you go to the dentist, and they are cleaning your teeth, getting rid of bacteria, that bacteria can enter the bloodstream and it goes to the heart, obviously, to get pumped around, and that bacteria in the blood can actually further damage the mitral valve and cause a heart infection. Who knew? So the American Cardiovascular Society or Heart Association, American Heart Association, I guess, and the Dental Association go back and forth and trying to figure out if patients who have this condition should take antibiotics before they go to the dentist to get their teeth cleaned, or if it's not worth it. And usually the recommendation changes like every year. So I've never actually taken an antibiotic before I go to my dentist. But anyway, so that's not the point. The point is that at this point, the blood is leaving the heart and it's leaving through these blue colored pulmonary arteries. Okay, so this is a pulmonary artery on the left. Okay, anytime you have an artery, it's carrying blood away from the heart. So this is a artery because the blood's going out of the heart. It's pulmonary because we're going to the lung and it's the left side. And then over on the right here, we have the right pulmonary artery. And I'm just going to abbreviate for sake of time, okay? At this point, the blood goes to the lungs. It binds to hemoglobin. Oxygen and hemoglobin rather bind together and picks up oxygen to come back to the heart. So when the blood comes back, it's going to enter through veins. Blood always comes to the heart through a vein. We have 2 on the left, and we have 2 over here on the right. These are known as the left pulmonary veins and the right pulmonary veins. Left, I'm just going to write PV, and right PV. Okay, when you watch the dissection videos, you'll notice that a lot of this stuff is missing from the hearts that they dissect, because meat markets and places that supply these things usually cut all of that off so that it's less bulky when they pack them together in the packaging. So a lot of times, like, all of this is not present on your heart. So you won't see anything but holes. It's kind of annoying. So I don't know quite what the video shows, but that might be obvious in the video that some of the stuff is not present. Okay, so then once the heart pumps the blood back in through those pulmonary veins, that blood is going to enter now the left atrium. And so that is going to be right up in here. Okay, the left atrium has its blood pool and then push open this next valve. And this is your bicuspid valve. Again, the picture here is misleading. It sure looks like there's more than 2 cusps or little bumps, but bi does mean 2. There is another name for the bicuspid valve. The mitral valve is its other name. Either name is fine. The blood pushes through that valve and should stay down here pooling in the lower chamber, which is known as the left ventricle. So we have left atrium up top here, and one of these labels is supposed to be for the left ventricle. And then once the blood pools in that left ventricle, it's going to need to be pushed big time out of the heart. And so this is a very thick wall of myocardial muscle here on the left side. Okay, gives a big push so the blood can go out. It passes through another valve. That valve is sandwiched right here. It's another semilunar valve. It's got that same crescent shape, and it's known as the aortic semilunar valve. It goes behind the pulmonary artery and pops out here. Pops out here. Here. Come on. My pen stopped working. Here. And then that blood's going to come up the aorta and come out through any of the holes that are options for it to move. There are different arteries that spin off of the aorta. There's actually hundreds of them. So for our purposes, we'll call this whole shebang the aorta. And then there's things like the brachiocephalic artery, which brings blood up to the chest. Okay, you have something called the subclavian artery, which brings blood through the neck. You have the carotid artery, which also could be found in the neck. That's the one you usually feel your heartbeat in, okay? Those are all different branches that are visible here. And then if you really pay attention, you can also see, hard to see in the picture, but there's an aorta that also drops down low. It's actually the same tube, I'm pointing it out down here. It's the same tube as the aorta up high. There's just a tube that comes down the back end of the heart. So you can't see it here in this picture, but it's back there. And that is the descending portion of the aorta. That's going to bring blood down to the lower half of the body. So your legs and everything like that brings blood from over there. Side note, when I did my first autopsy observation, the only one I've done, the gentleman who was being autopsied had been hit by a vehicle, and his heart had been pulled so that the top half of his heart went one direction and the back half of his heart went the other direction. And what actually happened is his aorta was split. So probably right around here, let's say, you can see where I'm marking, that descending portion of the aorta was ripped away from the rest of the aorta. So his blood had been coming up the aorta, ready to go out to the body, and it got completely ripped at that point. So a huge bunch of blood spilled into the inside of his body and killed him. He was also decapitated. So one of those two things killed him instantly. But that's a lot of blood. And so then in the activity, one of the questions was asking about the girth or the width of the artery versus the vein. And you probably saw, like, a vein is pretty skinny in its outer layers versus the arteries, by comparison, much thicker muscular wall, and they almost seem like they have a narrower opening. They don't really have a narrower opening. but they do have a much more muscular wall in an artery because arteries are carrying blood away from the heart. And with the exception of the pulmonary artery, that means that they're carrying blood really far. So they need to have a massive amount of push so they can bring blood all the way down to the very, very tips of the toes, tips of the fingers, and so on. And therefore, they need to be more muscular to do that, okay? One other side note was about mitral valve problems, mitral valve prolapse. We ask you to look that up in this activity. And one of the things that happens is if somebody's blood is not properly staying down here, because this mitral valve right here opens back up and lets blood squeak back up the direction it's not supposed to go. Again, the blood would squeak back up through here when it's not supposed to be. The end result of that, think through the next few steps. Blood is supposed to be pooling here and then going out the aorta. So if you don't have enough blood pooling, then two things will happen. One, you'll have less blood flow coming out of the heart with every heartbeat, which means less oxygen to the tissues, less nutrition to the tissues. So sometimes people feel like more tired, basically. They're not getting enough stuff out to their blood tissue or out to their body tissue. And then the second effect is that the heart is going to try and compensate. So it's going to try and push harder to get less of the blood that's pooling to get out. So this muscle right here might actually thicken, and that's known as left ventricular hypertrophy. And we had you guys look that up as well. The blood needs to have that much more push to get out if there's less of it, less volume. And so that muscle gets bigger as it has to work harder and harder. It's just like any muscle. If you work it, it gets bigger. So someone who shows signs of having left ventricular hypertrophy in like a ultrasound scan where you can physically see the heart muscle, an ultrasound would reveal that. And you would have an idea that something's going on that's causing the heart on the left side to work harder. Maybe they have a severe prolapse of their mitral valve. Maybe there's something else. Maybe they've lost elasticity in their muscle, so it can't contract the way it's supposed to, so it's trying to compensate and push harder. That happens when there's scar tissue in the heart, and that can be a result of a heart attack, for example. Sometimes if people have like really high blood pressure, then their heart has to work harder to try and get blood out into the surrounding blood vessels that are clogged with, artery clogging cholesterol, for example. One last note, and then I'm kind of done with the heart stuff for today. This middle section, does anyone know what the middle of the heart between the left and the right sides is called? You can unmute yourself if you know.
The septum?
Mm-hmm. So it's called the septum. And when babies are born, sometimes people are born with a hole in their septum. So imagine you have a little passageway right through the septum where blood from the left and the right sides can now commingle. Why would that be problematic? So the other night we had a Google Chat discussion that I led in a variety of directions because I wanted it to be like in class. I think Haley had asked the question. And so I don't like to answer everything directly, but I want you to think through the scenario. On the right side of the heart over here, you've got blood that hasn't yet gone to the lungs. So that's going to be blood that has less oxygen. Okay, we call it deoxygenated blood, but it's really just less oxygenated. On the other side, the right side, or left side rather, you've got blood that has already been to the lungs. So it's fully oxygenated blood. So what would happen if you have a hole? You're going to have the less oxygenated and the fully oxygenated blood that's mixing, okay? And that means that if this side is pushing blood out of the heart, it's not going to have as much oxygen in it as it should. So it has a similar effect as a mitral valve prolapse. The heart is going to have to work harder to try and push blood that much more powerfully. In addition, the heart might actually start beating faster. Just like, okay, we're not getting as much oxygen out, so let's beat faster and try and just get blood out faster. So people who have this problem might have what's known as tachycardia, where their heart rate is like boop, 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 trying to compensate. Usually it's a birth defect. So people who have this condition have it from birth. It's caught right away. Sometimes it's caught in utero, and they can go in and do a patch where they basically physically surgically insert a little patch to cover the hole. and the own heart tissue that's in that area, the muscle tissue, grows over the patch, and it's as good as gold. Okay, and then other medical dilemmas, you probably know a lot of stuff. We'll talk a little bit more about heart disease later and some of the interventions that are available for people who have other heart conditions, but one of those interventions is a valve replacement. So if one of your valves is just not working at all, to try and keep blood in one place and one place only, you can completely have the valve replaced. And a lot of times the valves are replaced with animal valves. So they'll use like a pig valve to replace, because the pig heart's about the same size as the human heart. Okay, so that's it for me, you guys. I'm sure there's stuff that I didn't cover that's related to both of these 411 and 412 assignments, but hopefully that was enough of a little preview if you haven't done the work yet. If you feel like you did something wrong and you want to go back and change what you've done for this assignment, feel free. I'm only going to look at whatever the most recent submission was. And I don't know if you noticed, but I did post a sample of student work. It was one of the first ones I graded, and it was done very well. And so I included that in the announcements section on My Connect yesterday, just so you could see like a sample of what I'm looking for. But please don't copy her answers. You got to find your own. Any last questions before we say ta-ta for the day, you guys? All right, when you're ready to head out, you can leave the meeting. I'm gonna stay in here for a minute in case anybody has any last questions before they head out.
Thank you.
You're welcome. Have a good day. I know it's Thursday already.
Thank you. I didn't even remember
Bye.
what it was.
I know it's Thursday.